In this video, I'm going to explain the general steps of tissue processing. Before our histological examination, we should do the following steps. First of all, we need to do the dissection of any subject, for example, any particle, any part of the body, human or animal. Then, after dissection, we use the machine to cut into our desired size and shape. I will show you this machine after my explanation from this page of paper. Then the second step is fixation. We use fixation and infiltration to stop the biological degradation processing and bacterial growth and put this tissue in actual state. So after fixation, we do embedding into any resin or paraffin wax. Then we mount it on a big slide. The step 5 will tell us the cutting area of excess tissue or resin. Step 6, we make section smooth. Then step 7, we mount on a small slide. After mounting the small slide, we use the laser diamond cutting machine into small slices for our histological examination. The rest of the slide or embedded material will go through these steps: the measurement, making the outer surface smooth by grinding machine or buffing machine. Then we start doing step 7. After laser cutting, we mount the slide on microscope, and if the microscope is not clear, then we do the measurement and cut the slice into small by using the grinding machine. If the required size has been reached, we use microscopy to record our experiment and look into the specimen, and we use to choose for the staining. Now let's move into the machines. This is the first machine we use to cut the big parts of our specimen into small size. This is EXKAT machine, and this is diamond cutter inside. After cutting into small size, our specimen will rest into different concentration of alcohol into that shaker machine connected with vacuum pump. It will take many days at different concentration of alcohol for infiltration, fixation and dehydration. This machine will be operated by these two knobs. This knob will show RPM, and this will give us the time duration. After dehydration, we use this machine for embedding. We put our specimen and embedding material, for example, any paraffin wax or some plastic material or some resin inside that boxes, and put our specimen, dip into it and lay them for long time. This is controlled by UV light and these knobs. After the embedding, our embedded material look like this. The specimen lie inside and we have this embedded material around it. Then we need to have this plastic slide. How to make the slide? We put the slide here and use these chemicals. Technovit 400 powder, 3 ratio, syrup 1, 2 ratio and syrup 2, 1 ratio. Put it on this plastic slide and then press it, connected with vacuum machine. After mounting on big slide, our specimen will look like this, but this area will not be smooth. To make it smooth, we use Technovit 7200 VLC solution, put it on it and then put it on this machine for several hours to make this surface smooth and repaired. After that repairing, our specimen looked like this, and we put it into the ventilated air to make it dry. After drying, we use this cover slip and this measuring tool to measure the thickness of this slide and thickness of our specimen. Then we need to mount this on small slide by using this machine and this Technovit 7210 VLC solution and connected with this pump. The pump will suck this slide upward and our specimen will lie down in this machine. We close this, turn the light on and wait for 8 to 10 minutes. After mounting on small slide, our specimen will look like this, sandwiched between these two slides. And then we are going to use this machine, the laser cutting machine. I already told you about this in my previous video, the separate video of this. And we use this cutting machine to cut into this very small size. After using the laser diamond cutting machine, our specimen will look like this. And then we again use this machine to measure the thickness of slide. If the slide specimen is thick, then we use this machine. This is called grinding machine, and this is a grinding paper. It is rough from outside. The specimen lies inside and it moves around this paper to make the specimen more thin. After thinning, we use this microscope to study our material. That's all.